right, let's go ahead and get started. Hello, everyone. My name is Ollie, the Technical Program Manager at Dovetail Genomics, and welcome to our Ask the Experts webinar, a practical guide to differential 3D genome analysis in the Dovetail Analysis Portal, aka DAP. Today's session focuses on how to move beyond individual 3D genome maps and begin extracting meaningful biological insights through comparative analysis across different conditions. We're joined by two experts from Dovetail Genomics. First, Dr. Myriam Elkhawand, our Head of Customer Success and Product Enablement at Dovetail Genomics, will provide an overview of the Dovetail linked read solution and the various epigenetic data analysis that we offer through the analysis portal, as well as key considerations in designing and structuring differential 3D genome studies. Following that, Dr. Jonathon Torchia, staff bioinformatics scientist of R&D, will walk through practical examples using the Dovetail analysis portal, demonstrating how to interpret differential features such as chromatin loops, TAD reorganization, AB compartment shifts, and more. Before we get started, if you have any questions during the presentation, please feel free to submit them using the Q&A button below. We'll address them as time allows at the end, and we're happy to follow up afterward as well. Without further ado, I'll hand it over to Myriam to get us started.
Thank you, Ollie. Let me share my screen from my end. And Ollie, can you let me know if you're able to see the slides?
Yep, looks great.
Okay, awesome. So hi, everyone, and thank you for joining us. The title of my presentation is Dovetail Linked Read Solution. So what is Dovetail Linked Read Solution? Dovetail Linked Read Solution is pairing the Dovetail Linked Read assay with a Dovetail analysis portal. So you go from sample to insights. You prep your libraries with a Dovetail linked read assay. You either prep them using our kits, or we prep them for you using our services. Then you sequence these libraries on short read platform. Then you access our analysis portal using credits or tokens. So you upload your FASTQ files, you submit your analysis through a few simple clicks, you get your results that you can download, explore using our interactive features, and also, you can export them at your end and use them for interpretation. So what that means is we're really trying to enable labs and researchers to go from their sample all the way to findings that they can interpret and get biological insights from. So what I'm going to do is spend some time talking to you about the different linked read assays that we offer, and then the different analysis services that we offer on the portal. And then my colleague John will go in much more detail about the analyses, especially the differential analysis, which is the one that we're releasing tomorrow. So the Dovetail linked read assays, they are all based on a core technology or workflow, which is the proximity ligation. So what you do is you first start with your sample, then you cross-link your sample to fix the chromatin, and that will preserve its 3D structure. Then you fragment, and when you fragment, what you get are those pieces of chromatin that are held in the 3D space. You ligate to create those chimeric DNA molecules. So what you're ligating essentially are pieces of the DNA or the chromatin that are not necessarily next to each other in the linear sequence space, but they were held in close proximity in the 3D space. So if you look at this example, you have this region and this region. They're not really next to each other in the linear space, but they are in close proximity in that physical 3D space. So what you do is you're going to ligate those two regions together. You're getting this chimeric molecule that's bringing together this region of the genome and this region of the genome because those regions were interacting in the 3D space. Following the ligation, you reverse the crosslinks, you degrade the proteins, you have your purified DNA that you then convert into a sequenceable library, and you're going to sequence your library on short-read sequencing platforms in paired end mode. So what you end up having are these molecules or these read pairs that are mapping to regions of the genome that can be distant on the linear sequence genome, but they are actually close to each other in the 3D physical space. So that's how you're generating your 3D genomics data. And what you would do with a standard NGS data is you would first ask, where do they map onto your genome? And that's exactly what you do with this linked read data as well. So you start with your FASTQ files, and what you have are, because you sequence as paired end mode, you have your read one, and then you have your read two. So you have your read pair file, you have your reference genome assembly, and you're going to align those read pairs to your assembly. And what you're going to get is, for example, you have this read one mapping to this region, and the read two mapping to this other region here. That's again not close on the linear space, but what that means is that this region of the genome and this region in the genome were interacting with each other in the 3D space. So you align all of your read pairs on the reference genome, and then you bin your genome. You can choose a bin size that's different bin sizes, 1 kb, 5 kb, 10 kb, and you're going to count those interactions. So in this example, you have these reads that are falling in this bin, and then they are interacting with this region, and you're counting 2 interactions. You have 2 read pairs that are supporting that indeed this region of the genome is interacting with this region of the genome. In another example, what this is telling you is that you have this region interacting with this region, and you have one read pair or one contact that is confirming or supporting this interaction. So one way to visualize all of these interactions that are happening would be to create this contact matrix, where it's actually telling you the different interactions that are happening across your genome. And you have the intensity of the color reflects the number of the frequency of contacts that you have. So if you look at this contact map, what this is telling you is that this region of the genome is interacting with this region of the genome, and you have a lot more interactions than, for example, you have here between this region of the genome and this region of the genome. Seeing the same thing just in a different format, you can also visualize those interactions as arcs. And this is telling you exactly the same thing. This region of the genome is interacting with this region of the genome. This region of the genome is interacting with this region of the genome. And you have more contacts here than here because that arc is higher. So the height scales proportionate to the number of contacts or the frequency of interactions that you have. So what you're really doing in this case, that would be more of that genome-wide approach. So you're looking at all the interactions that are happening across your genome, and you're trying to determine and identify or characterize the chromatin structure in your genome. How is your genome organized? We all know that genome is not randomly packaged in the nucleus. It's actually that folding or that packaging is very deliberate and it's highly regulated. That's what regulates gene expression. So what you're able to do with this 3D genomics data when it's genome-wide, or what we refer to as whole genome, you're able to identify the structure of the chromatin and how is your genome organized in your sample. So you're going to look at different levels of organization. You first have the chromosome territories. These don't interact much with each other. Then you're going to be able to say, how is your chromosome organized in A and B compartments? So A compartments would be your active compartments, where you have this open chromatin, you have a lot of transcription happening. And then B would be your closed chromatin that's inactive, where you don't really have a lot of genes that are being transcribed. Then you can also go with and explore the other level of organization, which is the TADs. So the idea with a TAD is that regions within a TAD interact more frequently with each other than outside of the TAD. And these TAD structures create sort of boundaries. So for example, if you have an enhancer and a promoter here within this TAD and another promoter here, the enhancer here will be more interacting and engaging with this promoter than the promoter that's outside of the TAD, because this TAD, again, these boundaries make it so that they're like insulators between the different promoter and enhancers. So you're able to identify the TAD structure. And then the other level of organization would be the loop. And that's really this point-to-point contact that you're identifying. It's that folding that's happening, bringing together two regions of the genome, for example, an enhancer and a promoter, in order to activate the transcription. So we have two assays that enable you to generate this whole genome data to interrogate the chromatin structure in your sample. We have the link prep assay and the micro-C assay that will allow you to do that. The difference, just as a very high level, is the enzyme that's used for the chromatin digestion. So with link prep, we use TN5. With micro-C, we use the MNAs. And then the library conversion is also different. You may have heard about our link prep assay, which is our latest assay. This is the assay that will really enable you to prep your libraries in a single day. So you start with your sample in the morning and you have the library ready to put on the sequencer at the end of the day. Micro-C, using the MNAs, is what's going to allow you to interrogate those interactions at an ultra high resolution. So if your research calls for any resolution more than 1 kb, or this nucleosome to nucleosome positioning, Micro-C will be the kit that's most fitted for you. So this is the genome-wide approach. We also have targeted approaches. So in this case, you're not really looking to interrogate and explore all the different interactions happening across a genome. You're specifically asking what are the interactions that are associated with either your protein of interest or your genomic region of interest. And we have two types of assay depending on that viewpoint. So if your question is, what are the different interactions or genomic regions that are interacting and are associated with my protein of interest, that would be the Dovetail high chip assay. So what you're doing here is you're cross-linking, you're fragmenting, but before you ligate your molecules, you do a chip with the antibody against your protein of interest. Then you ligate. So what you're doing here is first you are pulling down on that protein binding site, but also you're pulling down on whatever that region is that was associated or interacting with your protein of interest. So this is what you're going to ligate. And then when you start to map, what you're going to get is this is where my protein binds, and this is the region that's associated with it. So our Dovetail high chip assay will enable you to do that. Then after ligation, you'll convert that DNA into a library. And then again, you're going to sequence paired end mode on a short read sequencer. So this will be more from a protein anchored association or a protein viewpoint. Also, if you're interested in genomic regions, we enable that with our hybrid capture approach. We have a kit that's Dovetail pan promoter capture that's actually looking at all the promoters in your sample. And what you do is you first generate your library, then you hybridize it with probes that are targeting promoter regions, and that capture library is then sequenced. So again, here what you're saying is, this is my promoter region, and this is the region that's interacting or associated with my promoter site. So these targeted approaches are also enabled with our assays. And as I mentioned, we have the Dovetail high chip assay that supports this protein-centric view, and the Dovetail pan promoter capture that supports this genomic loci viewpoint. So again, this is the results that you're going to be getting in the schematic. With a Dovetail high chip, you know where your antibody is associated with your protein, you know where your protein binds. That's really what ChIP-seq will tell you as well. But what you're adding is this other dimension, and you're saying, my protein of interest is actually interacting with these regions. So for example, if this was a transcription factor, you're able to say that indeed this transcription factor is engaging with this gene and is engaging with this gene. And that's important because traditionally you would look at the transcription factor and you will look at which that promoter site or gene that's closest to it. But we all know that that's not necessarily true. The transcription factor can be acting on a promoter and a gene that's quite distant from it. So this will show you that physical interactions that are happening with the transcription factor and which actual gene it's engaging with. And from a Dovetail pan promoter capture data, what you're saying again is this is my promoter site, and I know that it's interacting with these regions. So you can look to see which are those regions, which marks they carry, are they enhancers, silencers, etc. So if you wanted to do those libraries, we offer our assays are accessible as kits and as services. So with the kits, you're going to get validated reagents, highly reproducible kits, user-friendly protocol, and you're going to get access to the support team. So if you have, questions mid-experiment, you can always contact us. Or you can actually have us prepare your library. So you send us your sample. Our experts in the lab will prep your libraries for you. And we have flexible configuration. So you can do the sequencing with us. You can do the sequencing at your end. We try to make it a la carte so that you really pick the service that fits your need. And then you also have access to project managers that will follow up with your project and make sure you're updated with where in the process your project is. So this is more from that library preparation side of things. What we're really excited about is that we've not only made those library preparation accessible, we've also paired them with access to the Dovetail analysis portal. So you sequence your libraries, you purchase tokens or credits that are very affordable, and then you get access to our analysis portal in order to submit your analysis and get your results. So we try to remove the bottleneck with data analysis, speed up the process of you getting to data that you can interpret. So we're taking on that data processing part of things. Few highlights of our portal and why we're really excited about it, and the users who have used it so far have also shared that it's simple to use. You're really submitting your analysis with few clicks. You don't need any bioinformatics knowledge or any coding or script. It's also very fast turnaround time. Some of the analyses will actually be done in a few hours. So if you're submitting high chip or a capture data, that's a few hours. Whole genome sequencing, if you're submitting that data, depending on the size of your files and the coverage that you have, it can take 1 to 3 days. So it's still very quick for you to get your results. And it's affordable. We price it very competitively to really enable you to process your data and analyze it. It's cloud hosted. It also has best-in-class tools and analysis practices. John will walk you through a little bit, our framework for approaching the analyses that's based on years of experience from our bioinformatics team. So we've incorporated those best practices into the analysis, and we use best-in-class tools that are all documented and well vetted. And at the end of the analysis, you're going to get a comprehensive report. So it will synthesize the findings for you. And that's what we usually recommend that you start with when we give you your deliverables. Start with a report to get this highlight of your findings, then go to your results. We send you all the files that you can download. We also have interactive features like IGB, like the contact matrix. So you can not only download your results, but you can view them on our interactive features. And all the outputs follow the industry standard output formats. So you can take those files and seamlessly integrate them in any downstream analysis you would like to do. So quickly, what are the analyses that we offer? We offer two types of analyses or two workflows, the epigenetic feature analysis and the differential epigenetic analysis. And these are available for human and mouse samples only. So the epigenetic feature analysis will support different data types. So for example, if you've done the libraries you prepped were the whole genome libraries, whether it's LinkPrep or MicroC, you're going to access the whole genome sequencing epigenetic feature analysis. And depending on what coverage you actually had for your sample, so whether 30x coverage, that means you prepped one or two libraries, the expectation is you're going to get AB compartment, stats, but limited loop calling because you don't have sufficient coverage. If you have 80x coverage, so you sequence to around 800, 950 million read pairs, and you prepped 3 to 4 libraries, you're going, again, to have access to the whole genome sequencing analysis. And here you'd expect to get AB compartment stats and in addition to loop calling as well. If what you prepared were the pan promoter capture libraries, so it's usually one library sequence 150 million to 300 million read pairs, you're going to have access to the capture epigenetic feature analysis. And what you're going to get here would be the calls or the loops that were called with Chicago. And if you have high chip libraries, recommendation is usually two libraries or one to two libraries with sequence of 300 million read pairs. You're going to get access to the high chip epigenetic feature analysis. And the results you would expect would be loops that are called with fit high chip. So all of this would be looking really at your sample or your experimental condition. But if you wanted to compare the chromatin structure between conditions or experimental setups, then you can also opt to add that differential analysis. So the differential analysis is offered as an add-on. That means you do have to first have submitted and have successfully completed the epigenetic feature analysis first for your samples. Then you'll add the differential analysis, and you can add it with either your whole genome sequencing capture or high chip feature analysis. So it's available with all three. And that will again be looking at the comparison of those chromatin structure between experimental conditions. To finish off a little bit about that differential analysis, and then John will add a lot more details. As I mentioned, it's available with the high chip capture and whole genome sequencing epifeature calling as an add-on. So you first complete the epigenetic feature analysis successfully, then you submit your differential analysis. It's going to allow you to compare between two conditions at one time. So we have it between your control and your test samples that you specify. And you're able to actually have one sample per condition or more than one sample per condition. So let's just go through all the different configurations that are supported. So again, you have two conditions, your control and your test. If you're working with whole genome sequencing, you can have one sample per condition. If you're working with capture, we recommend at least two samples per condition. So that one-to-one configuration will not be supported. If you're working with high chip, you can have one sample per condition. For the more than one sample per condition, the different configuration are 2 versus 2, 3 versus 3, 4 versus 4. So you actually now have, two replicates or more per condition. This is not supported today for whole genome sequencing. It is supported for capture and high chip. So you can design those experiments that usually call for replicates with replicates, and you can get your analysis done between two conditions with more than one sample per condition. A couple of questions here that you would expect you might have is whether you do need to have the same sequencing depth between your conditions. Obviously, you have your samples, your sequence, and it's rare that you get the same sequencing depth for all your samples. So the answer to this is yes, you can run the differential analysis. Our pipelines will perform normalization to account for any library-specific bias. So you can still run the differential analysis. And the second question, if you're thinking about it, is should my groups have the same number of samples? So should your control and your test have the same number of samples? And the answer to this is, while it's recommended, it's not required. So meaning that you can here have, two samples that are for control and three samples for your test condition. So it's quite flexible to account for a true setup of your experiment where you can't really control the sequencing depth. And sometimes a sample might fail the library prep. You might not have the same number of samples. So all of this is accounted for in the differential analysis that we are releasing tomorrow. I think with that, I'm going to hand it over to John, who will walk you through the analysis with more details. Let me stop sharing.
Perfect. Thanks, Myriam. Great. So what I'd like to do with the second half of our webinar is highlight some of the challenges that we've had internally as well as in the field for differential analysis, how we've chosen to address these challenges and solve them, and hopefully go through an example using one of the projects that we had internally in our lab, actually showing you what the types of analyses are available through the report. Before I do that, I'd like to say that in general, Hi-C analysis has come a significantly long way since it was first developed back in the early 2000, I think 2009. So it used to be something that was not really accessible to the greater research community. It was a highly specialized technique. There were the tools that were available were not really standardized or built yet. And significantly, the cost per sample was upwards of $10,000 to $20,000. So doing a differential analysis um, over 10 years ago was almost impossible. The last decade has made it a little bit more accessible. Cost has come down. Alternative methods have emerged, as Myriam kind of spoke about some of them. The first tools and kits were available, so you didn't need a dedicated bioinformatics team or a specialized molecular biologist to do your experiments. But still, your experimental design is still kind of limited because the cost is still prohibitive. And the tools that were available are still kind of in their infancy. Today, I would argue that this has changed significantly. We're now able to do high throughput kits. Link prep is one of those that allows for high throughput analyses. The workflows on just the general high C processing side are a lot more standardized now and available in the cloud as our analysis portal is one example of those. Really, it's opened the door to do these multi-sample, multi-condition, replicate driven experiments because the cost has come down significantly now on a per sample basis because of a lot of these technological advances. So it's really possible now, and I'd like to kind of highlight what types of analyses are possible. And I'm happy to say that there's a lot of great things that we can do with differential analyses. So Dixon, 2015 was kind of Dr. J. Dixon was one of the first to show that you can really learn a lot from how cell states change over time. And he showed this in a number of his papers, and I've just cherry picked a few of them from the literature. But types of questions you can answer are like, how does the chromatin reorganize itself during differentiation? So in this study, they looked at 5 different cell lines of various levels of differentiation states. And then using the AB compartments, showing that there's a switch from inactive to active components, and they're very specific depending on whether you're stem cell, you're a mesenchymal cell, or a pluripotent kind of neural cell. So being able to look at these on the chromosome level and say which genes are moving from active to inactive is really informative for things like stem cell differentiation. Going a little bit more granular to the TAD level, we're now seeing a lot more groups that are investigating things that are at the operon level. So hubs of regulatory networks, and this is just a cherry-picked example showing like the activation of a polycomb recursive network in their samples just by looking at the differences in TAD structures between their conditions. So if we're at chromosome level for AB compartments, we're at domain level for TADs. You can also do this, of course, at the chromatin loop level. So now we're asking gene-specific questions. Which genes control neural development? What are the enhancer promoter interactions that lead to the activation of transcription of my cell system? So this is just an example in mice, actually, showing the emergence of neural-specific enhancer promoters that are created. So lots of really interesting things that we can do with differential analysis. Although internally and in the field, there's been a lot of challenges for how do we actually do this. And I like to kind of not pick on, I love gene expression analysis, but gene expression analysis is an excellent comparator because it provides a good comparison of like the simple way to do differential analysis and why 2D or chromatin topology analysis is much more challenging. This table kind of highlights the main features here. So in gene expression, you're really only measuring one thing, which is the expression of a single gene across all the different genes. There's about 30 to 40,000 in the human genome. Probably not going to get much bigger over time if you include things like non-coding RNAs and the different types of regulatory RNAs. This might go up to 50 to 60,000 as we learn more. But in general, we're really stuck in that dimension of, in the sub 100,000 features range. Whereas for 2D topology, now this is where we've added like a multiplicative layer. So we're not just looking at the interaction of one locus, we're looking at one locus and how it interacts with every other locus in the genome. So we're taking basically one matrix and we're multiplying it by another matrix. And this can lead to billions and billions of pairs that we want to interrogate. So 30, 40 features for gene expression, billions of features for topology, which comes at a significant cost. The resolution of gene expression is fixed. It's one gene or one exon. Whereas topology, we have features across the various ranges of how chromatin is folding. So we have chromosome level. This is like your domains, your AB compartments. We have topological associated domain level. This is like groups of genes, or I like to call them operons, little transcriptional hubs. And we also have individual genes as well. So a lot of questions for the researcher. What level do I need to do my differential analysis, depending on the question that they're interested in? And there's also some normalization challenges as well. So gene expression, you only really have to normalize for library size. It's a relatively simple normalization. Whereas topology, there's a distinct distance decay model that you have to adjust for. So it's an additional layer of complexity. Closer you are in space, the more likely you are to be interacting with your neighbor. As you get further away, that likelihood of interaction becomes much, much smaller. So we weight things differently depending on where they are in space. Probably the biggest hurdle is really interpretability, right? So gene expression it's either up or it's down, or it's off or it's on. Whereas chromatin topology, it's not so simple. Something can be interacting, but it could be a repressive mark or it could be an activating interaction. So the interpretability is a real limiting factor for how do we take differential analysis in the 2D level and make sense of it. So again, not to pick on gene expression, the only real choices you have to make in gene expression are, am I going to use EdgeR or DSeq2? Just, the two main ones. Am I going to use FBCam or TPM? Very few changes. We've basically solved how to do it in gene expression. In topology, the researcher unfortunately has a lot more questions that they need to ask and to address their study of interest. So how do we categorize the types of differential analyses? I like to break them down into two main levels. We have the bin level. Bins meaning we don't necessarily care what the features are. This is feature agnostic, right? So we're basically looking at the individual interactions on a genome scale. And there's one-dimensional bins, which fall into things like TAD insulation scores, AB compartment eigenvectors, basically a bin that you can summarize in an individual 1D dimension. And 2D bins are basically where things start to they start to explode in the number of features, because now we're looking at every pair in your genome multiplied by every pair in the other genome, right? So when we do bin level types of analyses, we can get upwards of 10 to the 12 interactions that we might be interested in. So this really comes at a cost computationally and from an interpretation standpoint. Although the bin level analyses tend to be more sensitive, and I'll have a summary as kind of what the drawbacks are of the two different approaches later on. The feature level is when we're doing the comparative analysis after we've already called the individual features. Right, so a lot more simple to interpret from a biological perspective. It's a lot easier to compare just AB compartments between two samples, or compare the TADs that are present or absent in two samples. And the number of features generally stays, under the 10,000 to 50,000 range. So a lot more easy to handle, a lot more analogous to like what you would do for RNA-seq, where it's individual features that you're comparing. Although they come with trade-offs. I don't want you to read this entire slide, but the challenge of having these different levels that we can compare the genome, the bin level and the feature level, is that you have a number of different tools that are available to you, all with their own kind of strengths and weaknesses. So at the feature level, I just highlighted in green and red here some of the strengths and weaknesses. I'll have a better summary later. I don't want you to read this entire slide, but I just want to show you the sheer number of options that a researcher has, right? If they want to get highly sensitive, they want to detect every single interaction in their genome, they might want to focus on a 2D bin level type analysis. They tend to be the most statistically rigorous, and they're feature agnostic, so you're not limiting yourself to an individual feature. However, it comes at a cost computationally, because now we're in the billion feature level, and it limits interpretation, makes it a lot harder to interpret results when it's not a file that you can easily open in Excel, for example, or in something that you can, directly just read in a easier way. Um, they tend to be more sensitive, but also a little bit hard to interpret. So on the other side of the spectrum, the feature level, it's a lot easier to interpret comparing one feature to another. But again, that comes at a cost of sensitivity as well. You're not going to be able to get everything in that case, so that there is a real trade-off. There was a nice report last year actually that did a little bit of a benchmarking across the different tools. I'm not going to go over the whole paper, but they do highlight a few of the limitations that we see. Something that we've seen internally as well. But the challenges that the field is struggling with now when it comes to developing standards for this type of analysis. So what they were able to show, they looked at a number of different tools, feature level across bin level. And what this particular plot is showing that just the number of significant interactions varies widely depending on what tool you're using. One of them you can see in orange here just fails outright. So what this is showing is in the H0 setting, this is a setting where we don't expect any significant interactions. So it's really a measure of the false positive rate of an individual tool. Some of them just fail outright. So find, for example, you don't expect 20% significant interactions in the setting where there's no significant interactions. Others are highly conservative. So even down to 0% significant interactions. So you do expect, if your model is correct, to get some level of significance, even in the H naught setting, if you have a robust model. And others, they kind of vary the numbers, and they also vary across the different resolutions. Right, so there's a lot of inter-tool variability when it comes to detecting significant interactions. Another way to kind of highlight the H0 setting, again, this is a way to measure kind of the um, false positive ability of your test, or the ability to not call too many examples. This is a type of curve where you want this to be directly diagonal across the diagonal. So if you have a perfectly linear diagonal plot, it tells you that you have a really robust statistical test. And deviations away from that, so above that line, is a test that calls too much. Below that line is a test that doesn't call enough. What they're able to show that is that the replicate-aware types of callers actually perform really well in this setting, right? So in this case, the green and the yellow, they perform really well. They look like they're almost diagonal. And this is because it does a really good job at removing your inter-sample variability. But you'll notice for one of these, multi-HiC compare, that there's no yellow line. And that's because these types of multi-sample models come at a real computational cost. So when you go to a smaller chromosome where there's not as much data, for example, the tools start to just outright fail. Not enough data to fit the stringent models that they're trying to fit. Whereas the single sample, they kind of perform across the board, although they have some limitations as well. Some of them are they call too much, so the false positive rate's high, and some of them are a little bit too stringent. In the H1 setting, so H0 is a false positive measure. H1 is like the ability of the sample to actually of the test to actually detect the differences. Again, the main take home in this plot is there's a wide variability across the tools. No tool kind of performs um, the best in a different setting, and it also changes depending what resolution you're looking at. So all of the different models, they have their weaknesses and limitations, making it challenging. If you're the researcher trying to figure out how you should set up your differential experiment, this kind of confounds that. So the replicate aware seems to um, perform the best, although it does come at a significant computational cost. You'll see one of the multi-HiC compare just fails above a certain number of tests. It's not able to fit the model appropriately. And the other replicate-aware modelers, they come at a order of magnitude more computation time compared to the other tools. Here's one, HiC DC plus, like the more tests it's performing, it logarithmically changes the amount of time. So depending on what your question is, picking the right tool becomes much of a challenge. So just to summarize kind of what the trade-offs are between single sample, replicate-aware, feature level. The single sample way, this is like Hi-C compare and selfish. Um, their weakness really is in the false positive, and this is because it's not able to remove inter-sample variability, although they do perform really well at detecting any interaction that is there, it will detect it. Although a lot of that might be noise, so you might need some help in removing some of that noise and focusing on the biology that matters. So it's a good, this is a great method. And this is the method we use for our whole genome analysis because we are able to find everything, although we do need to go the extra step in limiting some of that false positive rate. Scalability is reasonable, so we are still querying millions of lines, but as you saw from the previous plot, the scalability isn't too bad when you're in single sample mode. Interpretability is still kind of a challenge because we're looking at still millions of lines, and the cost is relatively good because you're doing one-to-one comparison. So in order to do a whole genome comparison, you only really need one sample in each condition to get reasonable results. The replicate aware types like diff high C, multi high C compare, they perform the strongest statistically, right? So they have good performance in the false positive setting and good performance in the detection setting. Their scalability, though, is hit and miss. These are, I think, still quite limited tools, especially when you want to get to loops, which requires about 80x coverage. Sometimes the scalability there just becomes non-existent because these models require so much data. It's killing a lot of these tools. So it's good to see. We performed a number of tests before this benchmarking study came out, and we had the same experience. Some exam samples they fail and they just don't run properly. A little bit easier to interpret because we're reducing the false positives, right? So there's a lot less calls that come from these, and the ones that do come are higher quality. Again, comes at a significant cost. So now, in order to do a replicate-aware whole genome study, we have to get replicates for each sample. So there's a significant cost there. Whereas on the other end of the spectrum, the feature level, the main draw of doing a feature level comparison is the scalability and interpretability is much higher. It's a lot easier to interpret what it means when a TAD is absent or present, or when a loop is absent or present, although these are the weakest on the statistical front. So we're going to be missing a lot of variation just because we're focusing in on just the variance, the features that we're detecting. Right, so that is kind of the trade-off. This is an image that sort of summarizes that. If you're looking for robustness and completeness, you're going to choose a bin level experiment or a bin level tool. If you're looking for ease of interpretation at the feature level, you're going to probably choose a feature level comparison. You can probably see where I'm going with this is like, what is the best way to do this? And I would argue, and the authors of that study also argue, that the best way is to do both, is to actually operate on both ends of the spectrum so that you can get comprehensive results, but make sense of it by looking to the features themselves as well. So with the last part of the webinar, what I'd like to do is kind of show or highlight what it is we're doing for our differential analysis with a concrete example. So Myriam mentioned, what our portal is about. Basically, we can go all the way from feature calling through to interactive reports. So you do your feature calling, you can specify which samples you want to compare to each other. So feature level, um, bin level differential analysis, and then an interactive report that marries the two pieces of information that really helps with your interpretability. Um, so yeah, I'd like to kind of show you what it looks like. So this is a real life example of a whole genome experiment. This is a liver and a brain sample, healthy individual liver brain samples as a test experiment. Perform link prep. We perform through our analysis portal. What we got out of the end of it was about approximately 80x total coverage for the whole genome libraries. Now, Myriam mentioned you can't do multiple sample versus multiple sample for whole genome. That is true from a bioinformatics perspective, but what you can do is simply merge the data that you have, right? So in this example, we actually needed four replicates to get to 80x. These are technical replicates. if you have multiple samples, you can still do a differential analysis by simply merging the data. So this is an example of that. We had four replicates for a whole genome that equaled 80x. We were able to merge those together and perform differential analysis. This is a new plot that we've introduced really to visualize the whole genome differential analysis. So the most of you are probably familiar with volcano plots for differential. This is used extensively in gene expression where you have fold change plotted against the fraction of, or plotted against your significance. This is a lot more challenging when we're in 2D bin level space because you see what this number is, 18 million different interactions. So when you're at that level, a volcano plot becomes much harder to interpret. It becomes almost visually kind of uninterpretable. So we've tried to help you visualize basically the success of your differential analysis by introducing a volcano plot-like plot that has a third dimension, which is organized by distance, right? As we mentioned, like when you're in closer space, you're more likely to be interacting with one another. As you get further and further apart in genomic space, the chances of that interaction is much lower. So there's a difference in the number of significant interactions, or you expect the difference depending on where you are in the genome scale. So that's what we've tried to show in here. I'll just kind of walk you through. So in the solid line here, we have fold change, in this case, relative to the test sample. So it's the test sample over the control sample. 0 means there's no difference between your sample. If you're above that line, you have more interactions. If you're below, you have less interactions in your sample. So you see at the small distance or at the very close distance within like 1 kb of each other, there's no difference because those interactions are not considered significant. As we move on and on in the distance covered, now we see there's different fold changes. So that now we see differences between our samples. In the dashed line, we have the fraction of the bins that are actually considered significantly different, right? And what you can see is once you get to this range above roughly 10 to the 5, so 10 kb and above, this is where the majority of your biological variation is going to be considered significant, right? So we have a number of interactions. If you look at the dashed line that are specific to the liver, a number are specific to the brain, and the majority of the significant interactions actually occur in this space. So this is a nice way to sort of get a bird's eye view of how your differential analysis performed. If it didn't perform at all, what you would see is a basically a line that goes straight across 0, indicating no fold change, and you wouldn't get any significant interaction. So this dashed line would stay close to the bottom here, indicating that there's no significant interactions in your sample. It's just a really easy way to kind of get an idea of how your analysis performed. In this particular example, out of the 18 million considered interactions, about 145,000 of them were considered significant. That's still a lot. 145K is still a lot to go through. It's a lot kind of different than the couple thousand that you would get for a whole genome. So how do you make sense of that? And we think the answer is to marry that with feature level calls. So on the left here, we have basically those regions, the regions that were identified as significant, the p-values and fold changes associated with those. In this particular example, I have them sorted by positive fold change, which means there's more interactions in your test sample. And we've basically intersected that with the epigenetic features called at the bin level, or sorry, at the feature level, right? So the first number here is the number of loops, second number is the number of TADs. So for this region on chromosome 19, what this is telling us is that it's significantly overrepresented in our test sample because the fold change is positive. And we have 3 loops that are specific to the liver sample, which is our test in this case, and 2 TADs that are specific to the liver sample, one of which is, and there's another one that's concordant. So it gives you a really easy way to kind of shortlist your most significant interactions in a feature agnostic way with the features that you're more comfortable with interpreting. This is just a few examples. So you'll also get, you were to perform this through our analysis portal, you'll get the top 100 interactions at the matrix level. So you'll be able to visualize these really quickly and kind of flip through the ones that may be of interest to you. As an example of a loop that's specific to the brain. You can see there's no structure in the liver sample, beautiful loop created here. Actually 3 TADs in the brain sample. And one thing I think is really powerful about doing this in a feature agnostic way is we don't just get TAD and loop differences, we might also get structural differences, right? Here's an example of a SV-driven interaction. So this little inversion here, we know this is inversion just because of the way the matrix is oriented. The feature agnostic differential analysis was able to pick out this is something different here. Um, Even though it doesn't look like your standard TAD or loop. So in this case, we see that there's a loop in the brain sample. Now something gets mutated or there's an inversion in the liver sample. That loop is no longer there, and it's now some weird structural inversion. So it's a nice kind of byproduct of doing things in an agnostic way and then connecting it with the features. Although the feature level is still very important. So we also provide a feature level summary as well. So ignoring what we get from the agnostic bin level approach, we also provide what is the summary of the TAD loop and AB compartment changes. Right. So in this example, we do AB compartment analysis at 3 different resolutions, and we can quantify how many of those bins move from the active to the inactive component and vice versa, how many of them split and get merged into different sizes as well. We do the same for the TADs and the loops. We can quantify how many of the TADs are unique to our test sample, how many are unique to our control sample, how many of them go from a small multi-TAD region to a larger TAD hub. We call those merged. How many of them go from a large TAD hub to multiple nested TAD hubs? We call these split. And similarly for the loop changes, how many are unique? How many loops are present in both samples, but is strengthened in one relative to the other? And vice versa, weakened, and then also unchanged and concordant. Um, so that gives you a summary of kind of the feature agnostic, feature level summary. The last step is probably where the majority of our interpretation comes from. Um, it's still kind of challenging to take a list of interactions and associate those with genes. So what we're able to do here is take from what I just showed you, the different levels of differential analysis, we're able to take those regions, associate them to genes, and then perform um, a gene ontology type of analysis that is more analogous to what you would do for an RNA-seq expression. So yeah, we take those regions, we annotate them with genes, we perform gene ontology analyses for the genes specific to your test sample and for the gene set that's specific to your control sample. And what we're able to do here is show that there's a number of pathways that come up as being more overrepresented in one of your samples versus the other. So we provide kind of the highlights, the top 5. This is one of the first figures you'll see on our report. But also all of these results that I'm showing you come with the full table. So this is the expanded gene ontology analysis output. Shows you which group, which pathway, how many genes of that pathway are involved in the interaction, and of course the significance of those. And there's expanded output as well for in the portal when you go to download those results. So what I've shown you is direct snippets from the report. All of the underlying files that go into creating that report will be available for download as well. One of those expanded files is like this regulation hub, right? So this is an example. You look at the brain sample. These are all the genes that we identified significantly increased in the gene. You can see we get hubs now for brain development. We get different pathways that are related to the central nervous system, different cell cycle hubs. And in contrast, in the liver, we get a number of metabolic hubs that start popping up. Membrane lipid localization. I'm not going to start to claim that I'm an expert in any of these individual tissues, but at least kind of to the bird's eye view, to the untrained eye, we're able to really easily and simply bake down our results into the results that we think are the most important for your samples. So with that, I'm cognizant of time. I'd like to leave some time for question. There's no
Thanks for that,
perfect tool
John.
for differential analysis,
My
but by
pleasure.
combining bin level and
And
feature
hope
level, it
everyone have
allows
a
you
great day
for a more
ahead.
comprehensive and interpretable result. Everything I showed you is available through our portal, which launches tomorrow. Hopefully, I've shown that it provides a nice scalable solution for the full spectrum of differential analysis. So I'd like to leave some time for questions. Thank you.
Thank you so much, John, Miriam, for the great walkthrough. Now moving to the Q&A portion of the session. Feel free to submit any questions you have using the Q&A button below, and we're happy to take both technical or general questions. I'll give you a moment here. Also received a great question right here. And I'll go ahead and start with the question, and Miriam and John, feel free to jump in as appropriate. Um, so it's asked, how does the TN5-based link read assay work compared to micro-C, and what is the minimum cell number needed?
Maybe I'll take that one. So both workflows need 1 million cells. In terms of like the application, both workflows, link prep, that's TN5 based, and micro-C, will allow you to call your AB compartments, tabs, and loops. And they perform the same up to that 1 KB resolution. When you start to go below that 1 KB resolution, micro-C data allow you to call more loops because of the MNAs that's used for chromatin digestion or fragmentation. Now, in terms of the actual workflow, the first difference is transposase. TN5 is used for chromatin fragmentation and link prep versus MNAs for micro-C. But also for link prep, the library preparation is not your standard library prep. It's not the NGS library prep of, end repair, adapter ligation. It's a proprietary step that leverages having the adapters added during tagmentation. And that really is what enables us to reduce the library preparation and speed up the entire workflow for 1 day. So link prep is the only assay that will allow you to do library preparation in a single day. It also doesn't involve steps like biotin enrichment or biotin pull down that you'll see with a micro-C assay. So there are quite a few steps that are different. They both do the same thing in terms of calling AB compartments, SAD, and loops. The choice is really between what is the resolution that you'd like to interrogate your data at.
Awesome. Thank you, Miriam. Another question came in. What is the best method to examine promoter enhancer looping in small mouse tissues, such as mouse aorta?
Yeah, I think I can take that one. So I would probably point you, if loops are your primary interest, I would probably point you to the capture promoter. We have pan promoter capture for both mouse and human tissue. That's probably going to give you the most power specifically for promoter enhancer loops. If you're really interested in enhancers, then also the high chip for the K27 acetylation would be a nice kind of supplementary piece that would go with that as well. So if all promoters, I would point you towards pan promoter, specifically enhancers, then high chip with K27 acetylation is another option. Probably would shy away from the whole genome stuff there if you're really only interested in loops because you want power for loops.
Thank you, John. And for the second portion of that question, yeah, we are going to share a recording of the webinar. We already have your emails since you guys joined the webinar, and feel free to send that webinar link to your friends who may be interested in learning more on the 3D genome analysis stuff as well. Okay, let's perhaps we can wait a few more minutes for other questions. And if we didn't get to your questions today, we're happy to take any of them after the webinar. So feel free to contact us via email or by filling out the contact form on our website. We'll be happy to get connected. All right, if no questions, that brings us to the end of today's webinar. And on behalf of Dovetail Genomics team, I want to thank again Myriam and John for sharing your insights, and thank all of you for joining us today. I hope this session gave you a clearer view on how differential 3D genome analysis can help uncover some meaningful biological changes across different conditions and how you can apply these approaches in your own work. If you want to learn more, discuss your projects, or see a demo of our analysis portal, feel free to reach out to us again via email or filling out the contact form on our website. Thank you again for your time, and we hope to see you at a future webinar. Have a great rest of your day.